Assalamualaikum, beta. Today we are going to do the past paper questions of transport in plants. Let me share the screen with you, and these are all the questions from October November 2022 session. So let's start doing the past paper questions. So here's the paper which we are going to solve today. These are all the questions from the three variants of October November 2022, 11, 12, and 13. Let's start. The electron micrograph shows a cross section of a leaf vein and some neighboring cells, which structures contribute to mass flow by the use of proton pumps. So we all know that mass flow by the use of proton pumps, it is indicating the phloem and the phloem sieve tube and the companion cell. Companion cells have proton pumps which pump proton I outside. When their concentration increases inside the cell, they tend to move back to a co-transporter protein along with the sucrose, and then sucrose through plasmodesmata goes to the comes from the mesophyll cells, and then through plasmodesmata from the companion cell it goes to the sieve tube elements, and into the sieve tube elements water comes because concentration in the sieve tube elements rises. So water comes from the surrounding xylem vessels, and then it increases the hydrostatic pressure and massive flow of water along with sucrose. Sucrose takes place from the source to the sink. So that of course indicates this mass flow and proton pump is indicating towards proton phloem. So out of these, let me label these hollow tubes actually are xylem, and in leaves the upper side is always xylem within a vascular bundle of the vein. The upper one is the xylem and the lower one is the phloem. So this C is the phloem, which are not very large and not completely hollow, just like xylem. This B is the spongy mesophyll. And D is the lower epidermal cell. So it's so these are the four labelings, and the one which is responsible for that massive flow using the proton pump of the companion cell is the phloem. The diagram shows some type of tissue, some tissue types in plant organs. Within, that's the root where there's star-shaped xylem, and surrounding the xylem, within the arms of the xylem is the phloem. Then we have stem. Stem may towards the outer side is always the phloem. This is phloem. This number 1 is also phloem, and towards the inner side, it's always this one is the xylem. Leaf may, as I've told you, upper side is xylem, and the lower one towards the lower epidermis is the phloem, as we have just discussed. So keeping this in mind, 1, 2, 3 are phloem and 4 is xylem. Keeping this in mind, option A is the correct one. 1, 2, 3 xylem and phloem, and 4 is xylem. Which description of xylem vessel elements is correct? Xylem vessel elements. Cells join to form a tube, pits at intervals, because you know the plasmodesmatic connections within the xylem, within the cells which are actually specializing and differentiating into xylem, they are converted into pits. So there are pits at interval for the flow of water and minerals, sieve plates between cells. That's absolutely wrong. We'll not go through it further because there are no sieve plates in xylem vessels. Wrong. Contains cell joint end-to-end containing cytoplasms. That's wrong. There's no cytoplasm in xylem vessel. They're completely hollow. They don't have cytoplasm. They do not have, they have lignified cell walls only. They are all hollow from inside. There's no cytoplasm, no sieve plates, no end walls. Contains elongated cells with end walls broken down. As I've just said, no end walls, so they are broken down, located in vascular bundles in the stem and leaves. And in the Excuse me, in the roots, there is star-shaped xylem. In the stem and leaf, they are in the form of vascular bundles. So that is the correct option. There is no mistake in it. This is all correct description about what xylem vessel elements. Dead elongated cells, lignified cell walls with pits at intervals associated with companion cells is wrong. There are no companion cells associated with xylem vessel elements. They are with phloem sieve tube elements. So that's wrong. Option C is the correct one. Which properties of lignin are important for the function of xylem vessels in the stem of a tall plant such as tree? And properties of lignin. Lignin, you know, is a polymer of carbon and it is deposited within the cellulose cell wall, and it makes it rigid and hard and non-permeable. It becomes non-permeable because of the lignin deposition. Cell wall, when gets deposited with lignin, it completely becomes non-permeable, and it the structures inside, they die within some time period. They die because there is a surrounding non-permeable layer. So inside the structure, cytoplasm, nucleus, and all the organelles, they die after some time. That happens in formation of xylem vessel elements. So they are talking about the properties of lignin which are important for the function of xylem, which are important to its function. So let's see which properties of lignin are important to its function. It's inflexible, so does not bend easily. Correct. It gets so rigid. Cell wall is already rigid, but when it is when lignin is deposited on it, then it gets more inflexible and does not bend easily. It's not permeable to water, as I've just told you. It becomes non-permeable. Then otherwise, cell wall is fully permeable. But when lignin is deposited in it, it gets non-permeable. It is strong to resist collapse under pressure. Very correct, because you know, when we are drinking juice from straw and when we are drinking it very quickly, then even the straw, that shrinks and that contracts a bit because of the pressure of rising up of juice or water or whatever we are drinking using the straw. Same is the case with xylem vessels, that they might collapse, they might break under very high pressure of water when there is high rate of transpiration during the daytime and water is rising under a very high pressure. But these lignified cell walls protect it, prevent it, give it strength, and they prevent it from any type of collapse under pressure. It has weaker adhesion to water molecules than cellulose. That's incorrect, because lignin also has very strong force of adhesion with the water molecules. That's why, because of that force of adhesion, water rises up. So that is wrong option. First 2 and 3 options are the correct options. Which row is correct for the sucrose concentration and the water plants in a source and a sink in a potato plant at 12 p.m. रात के 12 बजे का वक्त, ठीक है, जिस टाइम पे ट्रांसपायरेशन और फोटोसिंथेसिस ये सब बिल्कुल ऑलमोस्ट they are not taking place at a very high rate. Photosynthesis का भी light independent reaction would be going on the Kelvin cycle, and जो आपका transpiration at that rate would be very low at night time, because there's temperature is low, there's less sunlight. So that's why when there are less activities going on, so they are talking about the sucrose concentration and the water potential in the source and the sink. So at night time, as all the activities are going at a very low rate, that's why the sucrose concentration within the source would be higher, as it's not moving around the plant at a very high rate. So it is at the source, and so sucrose concentration at the sink would be lower. And when the sucrose and sink sucrose concentration at the source and sink is high and low, then opposite would be the water potential. If source sucrose in the source is high, the water potential would be low. And if in the sink the sucrose concentration is low, the water potential would be high, because there would be more water. If there's less sucrose, water will be more. If there is more sucrose, water will be less, so lower water potential. So this you can easily do by doing the opposite of this one. And why have you made it higher and lower at night time? Because at that time all the activities are going at a low rate. That's why the sucrose, wherever it is, it will remain there, sink, and it is not going to be transported to the sink. because these activities are fast at daytime when transpiration is going on, when the xylem vessel elements are full of water, that wet water moves to the sea, so flowing sieve tube elements, massive flow is created, hydrostatic pressure is built up. So that's why this is according to this time period, we have made source and sink higher and lower, and we have made water potential opposite to it at this time of the day. Option B is correct. The diagram shows one possible way in which sucrose may be loaded into a sieve tube element. In which sucrose may be loaded into a sieve tube element. This is a companion cell. This is a sieve tube element, and this is a proton pump as being mentioned, which moves out, protons move out of the companion cell, so it's a proton pump. As a result of this, the concentration of protons will be higher over here, high proton concentration, they will tend to move backward from high to low. So they will move backward from high to low through a co-transporter protein, which is going to take protons, and along with protons, sucrose from the surrounding mesophyll cells will also be going against their concentration gradient into the companion cell. In the companion cell, this tree is representing the plasmodesmatic connection, that this sucrose is going through the plasmodesmata into the sieve tube element. So they are saying that which step requires ATP. Actually, here the sucrose is going against the concentration gradient, but it is getting the energy from the movement of protons. Movement of protons provides it energy to move against the concentration gradient. And protons are moving back because of this proton pump, which worked because of ATP. This is the proton pump, which is pumping protons against the concentration gradient out of the cell into the cell wall, and it is using ATP. All these processes do not need ATP. Protons are diffusing back, that's facilitated diffusion into the cell. Sucrose is actively transporting in from the low to high concentration, but that is secondary active transport, in which the energy of these proton pumps is being utilized for their movement going back to the these companion cells. And when the sucrose concentration is high in the companion cell, they will directly move from high to low concentration through the plasmodesmatic connection. So there is no energy in this process, no energy utilized in this process, but this process directly needs energy, that is ATP. So ATP is utilized by one only, this one. The electron micrograph shows a longitudinal section through part of a plant stem. Now you can see that these are very broad, sort of hollow, not completely, but slightly seem to be hollow cells, where it seems to be as if there are less organelles and there is less cytoplasm. And especially if we focus on this area, if I enlarge it, this area represents sort of sieve pores. Seems to be as if there are pores over here in this area. This helps us in recognizing it as sieve plate, and sieve plates are in sieve tube element. So this is a phloem sieve tube element, but they are asking about this neighboring cell to the phloem sieve tube element, and we know that phloem sieve tube element has the neighboring cell called as the companion cell, which gives it cytoplasmic support to survive and some other things as well. That's why it's called as the companion cell, which is helping it in its survival. So that is companion cell next to our phloem sieve tube element. Sodium chloride is added to a culture solution containing fresh water, single-celled plant cells. What happens to the water potential of the culture solution when sodium chloride is added, and what happens to the plant cell after 5 minutes? So we have kept sodium chloride in a culture solution, or उसमें fresh water single cell plant cells are also there. Now when we are going to add sodium chloride to it, what will happen to the water potential of the culture solution when sodium chloride is added? Water potential of the culture solution will, of course, will decrease. It means that it will become more negative. will decrease because you have added sodium chloride to it. So water potential will decrease and it will become more negative. And what will happen to the plant cell after 5 minutes? After 5 minutes, the plant cell is going to be plasmolized because it is going to lose water outside, because outside is highly concentrated. So plant cell after 5 minutes will be plasmolized and the water potential of the culture solution will be more negative. More negative means less water potential, less water molecules, less the water molecules, less the water potential, more the negativity. We will say that water potential is more negative. If we have two solutions, one is uh, said that is having water potential of -80, and if we say that the other one is having water potential of -20, so this -80 is having a low water potential, means it's a highly concentrated solution. And this one having -20 water potential means it has a high water potential. Its water potential is high. Okay. Which statements correctly describe transport pathways in dicotyledonous plants? Which statements correctly describe transport pathways in dicotyledonous plants? In the apoplastic pathway, water moves through the plasmodesmata. Absolutely wrong. In apoplastic pathway, water only follows and moves through cell wall, from cell wall to cell wall, and it just crosses the intercellular air spaces if any air space comes in between. So there's no plasmodesmatic connection because it does not enter the cell through the apoplastic pathway. It just goes from the outside of the cell, crossing the cell, passing through the cell wall to cell wall. And like if I draw these two cells, and this is the cell wall, then another cell, and this is its cell wall, then another cell, then this is another cell, and you can see there's an intercellular air space in between. So water is going to enter in, is going to pass from cell wall to cell wall. If it has to come to this cell, it is going to cross the intercellular air space and will come here into this cell wall, and then from here it will come here to this cell and then to the next cell wall. So it's not entering the cell. When it's not entering the cell, it is not going to pass through the plasmodesmata. So in apoplastic pathway, there is no cellular structure involved, only cell wall and the intercellular air spaces. So that's wrong. In symplastic pathway, water moves through intercellular air spaces. अब that's opposite. ये अब एपोप्लास्ट का लिखा हुआ है. Symplast में it passes through plasmodesmata, और epo में it crosses the intercellular air spaces. So that's actually opposite. Then the apoplastic pathway may be blocked by Casparian strip. The apoplastic pathway may be blocked by Casparian strip. Blocked by Casparian strip. That's correct. Because when it's following the apoplastic pathway, and if let's suppose in between comes the endodermal cells having Casparian strip, which are the bands of suberin, or sometimes they are completely suberinized. So when this water comes to this cell wall and when it comes over here, there is blockage of Casparian strip, that's the suberin band, so it won't be able to pass and follow the apoplastic pathway, that is only cell wall to cell wall. So it is forced to enter into the cell. This water is forced to enter into the cell. So when it enters into the cell, it has left apoplastic pathway and it has continued with symplastic pathway. So that is correct. The apoplastic pathway may be blocked by a Casparian strip. That is correct, and 3 only. Which statement about the movement of water from roots to leaves is not correct? Which statement about the movement of water from roots to leaves is not correct? Okay, keep it in your mind, not correct. From roots to leaves. A continuous column of water is partly maintained by the attraction of water molecules to cellulose, correct. Force of adhesion. Hydrogen bonding between water molecule enable massive flow of water, correct. That's force of cohesion. ठीक है, ये जो है ये adhesion को describe कर रहा है. That's the temporary hydrogen bond between the cellulose of the cellulose cell wall of the xylem vessel elements and the lignin of the xylem vessel elements and water molecules adhesion, and this between the water molecule the force of attraction because of hydrogen bonding is cohesion. Hydrostatic pressure in xylem vessel elements is higher in roots than in leaves. That's correct. Because of that hydrostatic pressure, this water then rises up in the leaves. So they are all correct. Water potential changes throughout the length of xylem vessel element. That is wrong. Water potential doesn't change. Once water rises up through the xylem vessel element, water potential should be higher. It should remain higher. So it continuously rises up through the xylem vessel element. If there will be differences in water potential, then this continuous channel of water is going to break. So there is no difference and there are no water potential changes throughout the length of xylem vessel elements. So that's absolutely wrong, and the wrong one is this D. Which terms describe the method by which water is transported within xylem vessel element? Co-transport is in phloem sieve tube element, where there is a co-transporter protein providing the way for the transport of sucrose and protons. Cohesion tension, correct. Within xylem vessel element, which is the method by which water is transported. So it's not even osmosis. When we talk about xylem vessel elements, then there are no partially permeable membrane. When there is no partially permeable membrane, it's not osmosis. Osmosis ke liye it's a must that there should be a partially permeable membrane. So when there is no partially permeable membrane, it's not osmosis. It's only cohesion tension. What's the main function of a companion cell in phloem tissue? What's the main function of companion cell in phloem tissue? Companion cells provide cytoplasmic contact with the sieve tube element for loading. That's correct. And that is by the help of plasmodesmata. Providing structural support for the sieve tube element, providing nucleus for cell division in the phloem, providing the source of assimilates for storage. They're all incorrect because they do not, that's not the main function of companion in cell in phloem tissue. The main is it helps in loading of sucrose, and it provides a cytoplasmic contact with the sieve tube element for the loading from the companion cells. Sucrose can go to the phloem tissue. The photomicrograph shows a section through a plant organ. Which statement could be used to describe this organ? The central region of this organ has supporting tissue. It is not supporting tissue actually. It is storage tissue. Pith है, ठीक है. So supporting tissue नहीं है ये जो है ये आपका सारा सेंटर में pith है जो कि एक storage tissue है. अच्छा. The endodermis tissue is a thick layer around the edge of the organ. So we don't see any endodermis over here. Thick tissue around the edge of the organ. Endodermis around the edge of the organ we can see, and epidermis is always there. There's no endodermis. Endodermis is only in roots. The epidermis tissue in this organ has extensions, trichomes. This we can see. You can see trichomes over here. Epidermis tissue in this organ has extensions called trichomes. You can see these trichomes. Trichomes are here. This seems to be correct. And then the xylem tissue is found in greatest density in the center of the xylem tissue. No. These are xylem. Here, the hollow tubes. But they are saying it's found in the greatest density in the center. No, center mein pith hai. That's wrong. The statements describe some of the properties of water. Some of the properties of water. Okay, water requires a lot of heat energy to evaporate, retains a lot of heat energy, is able to form hydrogen bonds with other water molecules, able to form hydrogen bonds with other polar and non-polar molecules. Which properties are also important for transport in xylem? Out of these properties of water, which properties are also important for transport in xylem? Yes, it requires a lot of heat energy to evaporate. So for the transport of water through xylem, it does not need to be evaporated, and evaporation does not have to play a role. That's involved in transpiration and cooling effect. It is helpful in leaving a cooling effect behind, but not for the transport of water. Retains a lot of heat energy. That is also not involved in transport of water through xylem. Like its temperature does not change easily, so that is not even involved. Is able to form hydrogen bonds with other water molecules. That's correct. That is involved in transport of water. Force of cohesion is able to form hydrogen bonds with other polar non-polar molecules. That's the lignified cell wall. That is also correct. Force of adhesion. These two play important role in the transport of water through xylem. That is D3 and 4. With xerophytic adaptations, reduce the water potential gradient between leaf structure and atmosphere. You know that the xerophytic plants, they have sunken stomata. They have spongy stem which stores a lot of water. They have deep roots, and there are different adaptations of these xerophytic plants, because they survive in very low temperature water conditions, where the water is very less in the soil, and there is very low humidity. So that is why there is high rate of transpiration, and there is a chance of wilting of the plants. So in order to conserve, xerophytic plants have a lot of adaptations. Some of the adaptations are mentioned over here, but they are saying that which adaptations are responsible for reducing the water potential gradient, like the difference in water potential of two areas. It means that those adaptations which increases humidity in the surrounding of the stomatal pore, so that there is minimum transpiration through the stomatal pore if it is humid outside. So under normal conditions, it's not normal, because xerophytic plants, they grow in very dry climatic conditions. But xerophytic plants have some adaptations, with the help of which in the surroundings of the leaves, they create a humid environment, so that there is less rate of transpiration. So that is a rolled leaf. When the leaf will be rolled, then it will, like if it's like this, then it will create its own atmosphere over here. In this atmosphere, if the stomatal pores are here, like we talk about marram grass is like that this is the upper epidermis and this is its lower epidermis, and it has stomatal pores. So the stomatal pores create a humid environment in the surrounding. So when humid environment is created in the surrounding, then there is very less evaporation through the stomatal pores, because it's already humid in the surrounding of the stomatal pores. So yes, rolled leaf is one of the way of creating a humid environment in the surrounding of the leaf. Hairy leaves, like having trichomes and having hair-like structures which actually trap water vapors and do not let them escape easily. So these water vapors also create a humid environment in the surrounding. Sunken stomata is like, it's like this, like over here, this is also sunken, which is you can say hidden stomatal pores. And this. So some of the leaves are of this type, where the stomatal pore is sunken, it's sinking, and there is a cavity in its surrounding, so that if water vapors are lost, transpiration takes place, these water vapors stays here in its surroundings, and they will create a humid environment in the surrounding of the stomatal pore. As a result, there will be very less rate of evaporation from the, is very low evaporation transpiration from the stomatal pore. So sunken stomata is also correct. क्या स्टोमेटा होता ही ऐसे ऐसे किसी लीफ में ना, ऐसे डिप्रेशन में जाके अंदर को करके स्टोमेटा होगा, ताकि वो अगर वहां से ट्रांसपायरेशन हो भी तो वो सारा वाटर वेपर्स इधर ही क्यूमुलेटेड रहे, और फिर उस ह्यूमिड इन्वायरमेंट की वजह से बहुत थोड़ा सा जो है वो लॉस ऑफ वाटर वेपर हो, ट्रांसपायरेशन का रेट खुद ही कम हो जाए. Fewer stomata is one of the adaptation of xerophytic plants, but that does not play a role in maintaining the or in reduction of water potential gradient. So Theostomata means less transpiration, but they are not able to produce this reduce this water potential gradient. So that's not involved. Fleshy leaves is also, it stores water, but it does not play a role in reduction of water potential gradient. So these three play important role, 1, 2, 3. Next is the diagram shows 7 stages of tulip bulbs growth. Which row identifies the correct stages in which the tulip bulb is acting as a source only or as a sink only? Tulip bulb is acting as a source only or as a sink only? Okay, source is from where the food is being transported to other areas, and sink is where the food is transported and received. So you can see that uh, if you see this one here, the tulip bulb is only acting as a source, like it's just providing it is not acting as a sink. It is not receiving food from anywhere. It itself is providing food to this growing plumule and this growing seedling, and it is getting energy and food from this bulb. So here it is acting only as a source. And if we talk about sink, where it is, where the bulb is not helping in making food and it's only receiving food, it's not providing food to any area, it's only receiving food from somewhere where food is being prepared, that is this one. That is source, only providing food, that is sink. So here these leaves are photosynthesizing, making up food, and that food is being transported to this bulb for storage for future use in future. So that's the sink, that's the source. Only, that's what they have asked. What would be changed if mitochondrial activity was inhibited by a metabolic poison acting on cells in the phloem tissue? Mitochondrial activity is inhibited. Mitochondria is responsible for aerobic respiration, for giving energy. So when there is no energy needing process will take place. So let's see which processes are energy needing. Concentration of hydrogen ion in the cell wall of companion cell would be changed. It's going to be changed because proton pumps are not going to work, and there will be minimum hydrogen ions which are going to be in the cell wall, and there are no hydrogen ions which are going to be pumped out into the cell wall. So the concentration of hydrogen ions will change in cell wall. It will decrease drastically. Concentration of sucrose in cytoplasm of cell in a leaf, that will also decrease. When proton pumps won't be able to work, there will be no sucrose in the cytoplasm of cells in the leaf. Okay, sucrose in the cytoplasm of cell in a leaf, like neither in companion cell there will be less sucrose, nor the process of photosynthesis will take place because there is no energy, and different functions will be inhibited because of the inhibition in mitochondrial activity, and as a result concentration of sucrose will also decrease. Hydrostatic pressure gradient in the phloem sieve tube. Hydrostatic pressure gradient in the phloem sieve tube will also change because mitochondria, again the same thing, mitochondrial activity is not going on, proton pumps are not working, sucrose is not being protons are not pumped out, sucrose is not co-transported, there is minimum sucrose in the phloem sieve tube element, no water is being transported from the xylem vessel elements into the phloem sieve tube elements, so there's no change in hydrostatic pressure, nothing will happen. So all these three will be affected because of inhibition in mitochondrial activity. By which process does sucrose move through phloem sieve tube element? Sucrose moves through phloem sieve tube element by massive flow. We know it's massive flow. Mass flow means many things dissolved in water going under very strong hydrostatic pressure difference from one area to another. That's massive flow, which happens in phloem sieve tube, and even in xylem vessel element as well, because xylem has water along with minerals and phloem has water along with so many assimilates, amino acids, ATP, nitrates, potassium, chloride ions, hormones, enzymes, all these things are going along with water. So it's a mixture of so many things. So that's massive flow. So in flowing sieve tube element, sucrose moves by mass flow. So these were all the questions from the October-November session 2022. All the three variants, 11, 12, 13 I have done, and these were the MCQs. Inshallah, I will share my next video on transport in mammals, and hope you are enjoying my videos. Hope you are understanding from these videos. If you are enjoying and understanding, do like it and share your comments and subscribe to my channel. Thank you. Allah Hafiz. Good luck.